All right, last video over this stuff. This one will be quick. This one's over DNA and then metabolites or folate, really. So here we have all our nucleotides, or nucleosides, I guess, that are phosphorylated. And they become deoxygenated by a ribonucleotide reductase. So it puts them in the deoxy form. And then they all push towards DNA, except the only one that isn't created is the thymine. So it has to be created from UDP. So UDP to deoxy. And then what ends up happening is the CDP, because it will push it into CMP, and then they have CMP deanimase. And deanimate it puts it into the uridine form of it. So from here, then we have the DUMP, and then it has the thymidate synthetase, and that's where you get this guy. And then it gets phosphorylated to be a tri, from a mono to a tri, and those all feed DNA. So that's how DNA is kind of made. Let's break it down. So we have DNA, then it nucleases, and it splits off into DTMP and DCMP. We'll talk about DTMP later, but DCMP, this is just like the other breakdown that we had earlier in the last video. So it has some non-specific phosphatase, puts it into deoxycytidine or deoxyuridine, and then from this it has the pyrimidine nucleic deanimase, and then we got to get rid of this deoxy. So what we do is we do a uridine phosphorylase, and we get uracil. So that's how we can break down DNA. And then on this side, because the thymine is a little different, it has to be broken down differently. So then it goes to phosphatase, and then we get the deoxythymidine, and then we have the uridine phosphorylase. It's the same thing on the other side over here. So now we have thymine, and now thymine becomes beta aminoisobutyrate, and that goes to succinyl-CoA, which is part of the TCA cycle, so then it gets kicked out into the TCA cycle. Okay, lastly, we have these folates, or anti-metabolites. They contribute to the creation of pyrimidines and purines. So I don't remember I'm sure you remember the videos, and it has the folate groups on it. This is where they come from. And then we also have the methylation of thymidine, which is important. So this is the cycle. So we have difolate, and then we have tetrafolate. We add a carbon in, and so that methyl group that gets added to the thymine, it comes from here. And so that's what turns the DUMP to the DTMP. It has just a little methyl group coming off on the structure itself. There's been a couple drugs that have been mentioned, like allopurinol, or what happens to, and that's helped to treat hyperuricemia. But these two stop the folate process, and they were specifically mentioned. So the MTX, and that stops difolate, so it kind of stops the cycle. And then 5-FU, it will stop DTMP. So this is kind of like a suicide DNA is what it was called. So this, I think, is anti-cancer. So this will stop you from having your cells replicate, which is what you want. So, all right, that's pretty much this module.